Good afternoon, everybody. My name is Shishir Shetty, and it's my honor to introduce the Osler Lecture. I'd like to thank Liz for the invitation. Liz, it's been an absolutely fantastic meeting, some really great talks. We had an inspiring George Griffin lecture yesterday, and we've got another keynote lecture today. I don't think I have to mention who William Osler is, but he did found the association. So this is one of the highlight lectures in medicine. And a lot of you know David Adams very well, so I don't really need to introduce him, and I can't really do justice to his research achievements. But for those of you that don't know David, he was the Pro-Vice Chancellor of the medical school at the University of Birmingham. He's an Emeritus, NIHR Senior Investigator, and Professor of Hepatology. He has started his career in Birmingham, but then went to the NIH in America and did some seminal work on leukocyte recruitment. He then returned to Birmingham and developed an internationally leading research group in immune-mediated liver disease and liver transplantation. And I want to finish on saying that he's been a champion for clinical academics and supporting their careers. And I have to say I've been very fortunate to be one of his students previously. So it's an absolute pleasure to invite David to give the Osler Lecture for 2026.
Well, thanks very much, Shish, and thanks to Liz and the organisers for inviting me. This really is a huge honour, and I'm delighted to be here. I'll talk a little bit about my career and how it developed and some of the concepts that we pursued during my career. And what I think is wonderful about this year's APAM is that we've got early career researchers and students here. So I'll try and drop a few pearls of wisdom that they might want to pay attention to as I go along. So I look back to when I first came to the Association of Physicians, and that was in 1988 in Belfast. And in those days, it was a pretty terrifying meeting to come to. You know, you only got invited as a junior if you were presenting a paper, and all the great and the good of British medicine were there ready to shoot you down. But if you survived, well, you know, you survived and you continued. And over the years, many people from my group, including Shish, have presented papers at the association, and it's an absolute delight to be back here today. If you can read the title, I was working on liver immunology even then, and I've stuck with that all the way through. So one of my pieces of advice to early career researchers is pick the right area to do your research in, pick the right area to specialise in. But of course we don't actually pick areas, do they? They tend to pick us. And how did I end up with liver disease and immunology? Well, the first person who really inspired me was this guy, Philip Gell. And again, the older members in the audience will remember the Gell and Coombs test, and they'll remember the Gell and Coombs classification of hypersensitivity, which was the first attempt to take immune processes into disease. And Philip Gell was the professor of immunology in Birmingham. He was a terrible lecturer. He strode up and down at the front of the lecture theatre, looking at his feet, never making eye contact, virtually no visual aids. But what he said was absolutely inspiring. And I'd come to medical school wanting to be a GP, not interested in all this science rubbish, but Gell really inspired me to think about immunology, and that's stayed with me the rest of my career. And then the other person who, again, people in the audience, some will know very well, who got me into hepatology was Elwyn Elias. He had just come to Birmingham from the Royal Free to set up the liver units in the mid-80s. And I wanted to do gastroenterology, and somebody said, go and see Elwyn, because he's got good connections in London. He can help set you up with a fellowship in London. So I went to see Elwyn, and much to my amazement, he said, well, I'll offer you a fellowship here with me, which I thought was fantastic. Except what I didn't know was that Elwyn had no money and basically no ideas and no lab. But Elwyn being Elwyn, it all got sorted out, and the rest is history. And that's how I ended up working in liver disease. And it's been an amazing field to be in over the last 30 or 40 years. You know, we've had new diseases. One of those new diseases, hepatitis C, we're about to try and eradicate from the UK. It's incredible in my career time. And even a disease like non-alcoholic fatty liver disease, you know, when I came into hepatology, there was a big debate as to whether fatty liver disease was just a pathological observation or whether it had any clinical relevance at all. Well, now we all know, of course, that it's the dominant liver disease that we all see. So a fantastic clinical discipline to be in. And this was a study that really woke people up in the Lancet in 2006, showing the mortality from liver disease in the UK and Scotland, and a really sharp increase, and not all due to alcohol, but due to a range of liver diseases. And if you compare on the right-hand side of the graph, mortality from liver disease, whereas many other chronic conditions, mortality is improving, liver disease continues to get worse. And we don't have treatments for most chronic liver diseases, and that's something I'll talk about in the rest of the lecture. The one treatment we do have is liver transplantation, which again, in my career, has gone from being a controversial experimental treatment done in very few centres to being a globally available treatment for end-stage liver disease with fantastic survival rates. But really, we should have something other than liver transplantation to treat all these patients with chronic liver disease. So during the same period of time, of course, the advances in immunology have been massive. We really understand immune processes and immune regulation far better than we did. And of relevance to my talk, particularly the understanding that tissue microenvironments are so important in allowing the immune system to drive diseases and offer the opportunities to translate that into therapy. So two areas where immunology has linked up with parts of liver science that really didn't think there were anything to do with immunology. One is fibrosis and cirrhosis. And the work here led by several groups, particularly my good friend John Iredale, who's done fantastic work throughout his career, showing how dysregulated immunity and inflammation is an absolute critical driver of fibrosis. And making that apparent is so important because now we've got another string to our therapeutic armory. We can look at immunotherapy as well as just trying to reverse fibrosis. So John's work has been seminal in that area. And another mate of mine, Stuart Forbes, demonstrating how inflammation and immune responses are so important in driving the liver's ability to regenerate. And we all know that the liver is probably the only organ that can truly regenerate. So there's been an understanding over the years, a link between immunology and some of these processes which were considered to be completely distinct in the past. And indeed, the work that Stuart's done with Phil Newsom has led to stem cell trials to see if you can actually promote that healing inflammatory response in patients with cirrhosis. So, coming back to immunology, another realization over the last few decades is that actually most of the immune reactions that take place in our bodies are there to suppress immunity and inflammation, not to stimulate it. And this is really important for the rest of what I'm going to talk about. So when the immune system is activated, of course you need an effector arm to clear that virus, but you then need a regulatory arm to turn everything back to normal and stop that inflammation becoming chronic and perpetuated. And it's this balance between regulation and effector arms which is so important. And in the context of what I'll talk about, we'll talk later about these cells called regulatory T cells. So the three autoimmune liver diseases that we deal with are shown on this slide. And again, interesting, when I came into hepatology, autoimmune hepatitis was known to be an autoimmune disease. It's classic, it's associated with other autoimmune diseases, affects women, responds to corticosteroids. PBC and PSC were not considered to be autoimmune diseases. And it was the work of Howard Thomas at the Royal Free that really began to make us apparent that PBC might be an autoimmune disease. And then the work of Maggie Bassendine and Steve Yeaman in Newcastle when they defined the autoantigen, the anti-mitochondrial antigen back in the mid-80s. And it's taken even longer for us to appreciate that primary sclerosing cholangitis is an autoimmune disease. And just at the end of last year, there was a fascinating paper in Nature Medicine showing that a really strong T and B cell response against the Epstein-Barr virus is strongly associated with PSC. So these three are autoimmune diseases. What's really interesting, we'll come back to this, is that autoimmune hepatitis focused on the hepatocytes in the liver responds to immunosuppression. PBC and PSC focused on the bile ducts don't, and that's a real conundrum. So what are the unknown questions around autoimmune liver disease? Well, the triggers of autoimmunity are still not well understood. That's not my area of expertise, so I'm not going to talk about that. But this question about how do you end up with chronic inflammation, which is activated and self-perpetuating? Why and how does this happen? Why do some diseases fail to respond to immunosuppression while others do? And how understanding these processes might lead to new therapeutic approaches. So I'm now going to give a really, really simple cartoon of immune response, and I apologize to any immunologists in the audience. But basically, the immune system works by generating naive T cells from the thymus. Those naive T cells recirculate through secondary lymphoid tissue, and if they encounter a dendritic cell coming from tissue with an antigen that they recognize, then those T cells are activated, and you end up with effector cells that not only have activation potential, but also express molecules that take them back into the tissue where the antigen sits. And then those cells become long-lived memory cells. And it's this part of the process that I want to focus on. I spent most of my career, as Shish was mentioning, trying to understand the factors that regulate the recruitment of effector T cells from the blood into tissues, and of course particularly the liver. So lymphocyte recruitment from blood is critical for the development of immune-mediated disease. Again, remarkably, 40 years ago this wasn't fully accepted. It's quite incredible really when you think about it. I was very fortunate that in 1991 I went to work with this wonderful man, Steve Shaw, who really should have got a Nobel Prize, but he was far too modest. And Steve was working on the adhesion cascade, which we're all now fully familiar with, this idea that you take a flowing cell from the blood into tissue via sequential interactions with the endothelial cells that line that tissue. But this was considered to be a highly controversial and unprovable paradigm. And Steve and others, Eugene Butcher and Tim Springer, show that this really does work. And it's totally opened up the whole concept of lymphocyte trafficking and homing. So it was a wonderful time, and the stuff that I learned with Steve has sat with me through the rest of my career. So the next point for the early career researchers, if you're thinking of doing a postdoc, think very carefully where you do it and who you do it with. And working with someone like Steve was just an absolute privilege. So when I came back from Steve's lab, we'd helped to define some of those mechanisms, but it was very much the early days. And I came back as a hepatologist, and I really wanted to see if any of the principles we've been studying applied in the liver, and whether the liver might have slightly different molecular signals for recruitment than other tissues. And the reasons for thinking this is that the liver has a unique vascular bed. It's a low-flow vascular system. The sinusoids are lined by phenotypically distinct endothelial cells, and the requirements to take a flowing cell through that vascular bed were likely to be different. So we set up to define these different steps in the adhesion cascade over many years, and we and others, but we made, I think, a major contribution to this whole concept. And this just shows the complexity of the process. Some of the molecules involved are involved in other tissues, but I really want to focus on two molecules. Well, I want to focus on one molecule, but I'm going to mention another molecule, which were distinctive and different from most of the molecules involved in other tissues. And the molecule I'm not going to talk about is stabilin-1 or clever-1, and this is the molecule that Shish works on. This is a fascinating molecule, and this appears to regulate the recruitment of suppressive regulatory cells into tissue. And therefore the work that Shish is doing is looking at targeting this receptor to drive immune responses in cancer, a novel approach to trying to activate immune responses in cancer. But I'll leave Shish to tell you that story. So the molecule I want to focus on today is vascular adhesion protein 1. And this was discovered by Marcus Helmy and Sir Pielken in 1992. Then they published a paper in 1993 showing the distribution of VAP1 in different tissues. And this is what piqued my interest, because when you looked at tissue, the one tissue where it shone under normal conditions was the liver. Most other tissues, it wasn't expressed. So I thought, wow, that maybe this is the liver homing receptor. So I contacted Sirpa and Marku, and they provided us with reagents to look at VAP1. And this has been a 30-year collaboration. This collaboration is still going on. And this is another message for the early career researchers. Collaborate, 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 and make sure you collaborate with great people, with great ideas. You'll make great friends. The picture here is of a scientific retreat in Finnish Lapland, which was brilliant science, great fun, too much alcohol being drunk. But collaborators are wonderful. So pick the right collaborators. And I just want to mention 3 people here who really helped me in this collaboration: Chris, Shish, and Trish. So this is what VAP1 looks like. And when we started working on it, we thought it was a different type of adhesion molecule, but we thought it worked by binding through sialic acid residues to lymphocytes. And indeed, it does that. But when it was cloned, we had a massive wake-up call, because this is a cell surface enzyme. It's a semi-carbside sensitive amine oxidase, and it catabolizes amines to generate aldehydes, ammonia, and hydrogen peroxide. And this was a complete surprise. But it turns out that that activity at the endothelial cell surface activates a whole string of pathways, including NF-kappa B activation, changing the endothelium to express different patterns of receptors and to facilitate leukocyte recruitment. So for the first time, we've got an enzyme that was promoting recruitment at the endothelial surface. And when we began to look to see what were the bacterial amines that could drive this process through VAP1, many of them are bacterial amines, and in particular, and this is work of Palek Trifedi, these are amines that are produced particularly when you get dysbiosis, that's dysregulation of the gut microbiome. So you can imagine that in bowel disease, these amines are produced in greater abundance, particularly amines like cysteamine. They go up the portal tract into the liver, they can activate VAP1 and promote leukocyte recruitment into the liver, hence explaining in part why we have this strong link between the gut and the liver. So a fascinating molecule, a molecule whose enzyme activity appeared to be important, and this just shows a recent review showing all the different places where that enzyme activity might operate in the adhesion cascade. So we became really excited, and we published some work 10 years ago now showing that this molecule promotes fibrosis and inflammation in animal models as well as being expressed in human liver disease, and moreover using enzyme knockout versions of VAP1, showing that the enzyme activity is really important for that function. So we became really excited, and we set up collaborations with industry partners to see if we could get hold of inhibitors to this enzyme. And people had been studying the enzyme because SSAO is important in the regulation of the insulin receptor as an intracellular enzyme, but nobody had thought of it as an anti-inflammatory target. So we got very excited about this, and we worked in particular with a company in Australia, and we shared our data on liver disease with them. They got some interesting reagents, which they let us have. And then a few months later, we saw this. So Boehringer Ingelheim had bought up their whole program for $750 million to allow them to develop these agents as treatments for liver disease. Well, it's great for liver disease, but it wasn't quite so great for us because we hadn't protected our IP properly. And so my next lesson to the early career researchers is if you discover something that's really novel and looks interesting, get your IP protected. So anyway, I think what's interesting about this whole story is this whole concept of bench to bedside. We talk a lot about this. But, you know, we first reported a role for VAP1 in liver disease in 1996. The first clinical trial was published by Gideon Hirschfield, who started that in Birmingham in 2024 using an antibody to block VAP1. Unfortunately, it was safe, but it didn't work. And then the Boehringer Ingelheim enzyme inhibitors were published in Nature Comms a couple of years ago, showing that they do have an effect on both inflammation and fibrosis and suggesting it's an attractive target in liver disease. So that's 30 years, and we've not even got this into the clinic. We've just got it into clinical trials, so a very long road. So for the final bit of the talk, I'd want to come back to this idea of why don't PBC and PSC respond to immunosuppression? And I think the reason lies in this histology of the disease where you see the bile duct at the center of this intense inflammatory infiltrate. And these inflammatory infiltrates become organized into what's called portal-associated lymphoid tissues, a highly organized lymphoid aggregates focused on the bile ducts. And if you look at the cells surrounding those bile ducts, they are effector cells, particularly cells, for instance, expressing IL-17, which is a really important effector pathway in damaging tissue. And if we take these cells out of the liver, they express a receptor called CCR6, a chemokine receptor, and the bile ducts in the liver express the ligand for CCR6, CCL20, in tissue. And then this was work done by Yeu in the lab. We were able to demonstrate that if you look at co-cultures of these different cells, that the cholangiocytes are able to produce this chemokine which attracts Th17 cells in and locks it into the bile duct. But even more than that, the Th17 cells produce cytokines that amplify that production of CCL20. So we have an autocrine loop that is emphasizing the inflammation in the tissue. And of course, this brings up some therapeutic options, but again, targeting ustekinumab so far hasn't been effective. And the anti-CCL20 drugs generated by GSK don't really seem to have gone anywhere, but some opportunities there. So this is work done by Ye. And then the work becomes even more interesting, because if you look at what these cholangiocytes are producing in response to these effector cells, the cytokines they produce, if you expose them to regulatory cells, the cells that are there to suppress the inflammation, convert those regulatory cells into effector cells. So you can begin to see how this microenvironment is really hostile to regulation. Everything is driving things towards the effector arm. And this linked up with some lovely work done by Adrian Hayday around the same time, which he called the epi-immunome. So he was able to demonstrate in several systems that if you damage epithelial cells, the initial response is to produce factors that drive repair of those cells. But if that damage persists, increasingly you get the production of factors that drive inflammation, and particularly drive IL-17 driven inflammation. And once again, if we look at these factors, these are all cytokines where we now have drugs and tools to try and address them. So this activated stromal compartment, although highly organized, should hopefully be susceptible to intervention and treatment. So why is it that in autoimmune liver diseases the results have been so disappointing? Plenty of targets, but very little success. So part of this is due to the reluctance of pharma drugs to trial drugs in liver disease. That is beginning to change now. Part of it is due to the chronic course of diseases and the fact that markers of response are difficult. And I think this organized inflammation with this activated epithelial stromal part is absolutely crucial. And the tissue microenvironment favoring persistent inflammation over resolution. So that's a very depressing end to the talk. So I wanted to show a few more slides to try and offer a little bit of hope. So one approach, instead of attacking those pro-inflammatory, destructive cytokine signals, can we actually boost the regulatory side of this whole process? And you'll all be aware, I'm sure, of the work of John Todd in Oxford showing that you can use low-dose IL-2 to boost regulatory T cells in diabetes, type 1 diabetes, with some effect. And several groups have been looking at the possibility of T-reg infusions, and Giovanna Lombardi and Robert Lecture set up a unit at King's to generate these kinds of regulatory T cells in the context of immunotherapy and liver transplantation. So what about if we infuse these T cells, where will they go, and will they just be killed by this hostile environment I've been talking about? Well, Ye again, working with a scientist, Stuart Kirbishley, set up ways of isolating regulatory T cells from patients with autoimmune liver disease and getting them to clinical purity, and enough cells that we could then label them and inject them back into patients. And this is where those regulatory T cells go. This is a patient with autoimmune hepatitis. And if you look on the left, you can see the liver lighting up with these infused regulatory T cells. So the cells go where we want them to go. They go back to the liver, which is great. But can they survive and can they function in this hostile environment? Well, to try and answer this question, Ye is working with Shimon Sakaguchi, and I hope you're all aware that Shimon was awarded the Nobel Prize for Medicine in 2025 for having discovered regulatory T cells. And I'd just like to point out that we were ahead of the Nobel Committee because we offered him an honorary degree in 2019. So that's me wearing a very silly hat. So Shimon has been working with Ye for several years now, and I'm absolutely delighted to say that I think there's a real possibility they might be doing something very exciting. Because Shimon has developed ways of manipulating conventional T cells and converting them into Tregs that not only are stable and functional and able to be rapidly expanded, but are not inhibited by immunosuppression and can survive in hostile environments. And Ye has shown that these Tregs express the receptors required to get them into the liver. So the Wellcome Trust have just given Ye and Shimon, together with Graham Anderson here in Birmingham and Kelly Dendrew in Oxford, a large grant to really try and take this through as a novel therapy for autoimmune liver disease. So really, really exciting stuff. So finally, to finish, my last piece of advice to the early career researchers: recruit great people, because most of the stuff I've spoken about today has been done by fantastic people who've come into the lab, and I've just taken the credit for it over the years. And then one very final piece of advice to you all. I'm the registrar at the Academy of Medical Sciences, and the Academy is co-sponsoring this meeting, and there are a couple of programs which may be of real interest to early career researchers in the audience. The mentoring program at the Academy is fantastic. It can link you up to a fellow who can offer you really great advice around mentoring. And for people a little bit further on, there's a program called Flyer, which is an 18-month program that allows emerging leaders to work between sectors. If you really want to try and work across sectors and drive advances in research and healthcare, this is a wonderful program, and your president, Liz, is an alumna from that program. So if you're interested, have a look on the Academy website. Two great opportunities. And on that, Shish, I will finish. And thank you all very much.
Thanks, David, for telling us your story. Obviously the challenges, but really some inspiration there as well. Thank you very much. And I'm going to ask our president to come up stage to David. Don't run away.